All right, so these are our protein synthesis notes. If you do not have them open yet, they are in Unit 5. You want to go ahead and open your copy of the Cornell notes here and make sure when you're done you do highlights and a 3 to 5 sentence summary, and then create a link on page 28 of your notebook. All right, so let's go ahead and get started. Okay, so protein synthesis. So it is the protein making process. So what is our essential question? How does DNA code to produce proteins? Proteins are used all over our body and our muscles, but they also make up the color in our eyes. They make up our skin. Everything you have, all your traits, your hair, everything, color of hair, is done by proteins, okay? So we're going to learn how DNA, now that we know how it's replicated, how does it code to produce those proteins and make it happen? All right, the whole theory that we call all the way from DNA all the way to those proteins, we call that the central dogma theory. And so it's about how that all works together. So the central dogma is how DNA goes to RNA, which we'll learn what that is in a minute, and then finally to a protein. And it consists of 2 steps we're going to talk about. It consists of transcription, and then translation. So it explains how DNA codes for proteins. So if you think about transcription has to happen first. Let's say you have a cookbook, it's in a different language. It needs to be transcribed into English so that we can actually understand it, and then it can be translated into something that we can actually eat, okay? All right. So proteins make up all living things. So why do we even care that DNA makes up proteins? Well, proteins make up our hair and nails, our blood, our brains and nerves, our enzymes, cellular construction workers, so all those things that they build cells out of, antibodies, so to help us from being sick, cellular messengers, they send messages so that my fingers work and my toes work, and then also muscles. So you can see if we didn't have proteins, nothing would actually happen in our body, and we couldn't think, and we wouldn't have any blood. We probably just wouldn't even be alive, okay? All right, so one of the things we can start with, this is kind of a 3D model of what it looks like. So what we're going to learn is it's going to make a chain of amino acids. Remember, amino acids are the monomer of proteins, and then they will come together to make that polymer, which is the protein molecule, okay? So proteins are actually composed of amino acids. Those are our monomers, and there are 20 different ones, and different combinations can do different things. So different proteins are made by combining those 20 different ones in so many different combinations. So if you figure out, I mean, we're talking millions of different combinations to make all the different things we need: eye color, hair, blood, muscles, okay, heart, organs. They can be combined to create those. And then proteins, actually what we'll talk about before we get there, but proteins are manufactured or made by those ribosomes. Remember when we talked about all those organelles? Well, now we're going to talk about something. So it's ribosomes. Those make the proteins. Those actually construct them. Remember when we saw the endoplasmic reticulum, we had the rough one and we had the one with all of those little dots. Those are the ribosomes. So those little tiny things put these amino acids together and actually make our protein. And we're going to learn how that happens. Okay. The functions of proteins. Remember, let's just remember what the functions of proteins were that we talked about last semester. They help fight disease. They build new body tissue. Enzymes are used for digestion. Other chemical reactions are proteins. Remember, they can speed up the rate of reaction. We talked about enzymes last semester. And then, of course, they are a component of all the cell membrane. So that's how our cells keep everything together. Okay. All right. So let's talk about the first step. Step 1. It's called transcription. Okay. So making a protein or transcription. Our first step is copying of genetic information from what we call DNA to RNA. RNA is what the full name is ribonucleic acid. So it's a different form of a nucleic acid. And we call it ribosomal because now it's going to head to the ribosomes and make that protein. So RNA is not an exact copy of DNA. It's a translation of that DNA into something to make proteins, okay? Or transcribe to translate, okay? And it's called transcription when we go from DNA to RNA. We're going to make it a totally different kind of molecule. Not too different, though, you'll see in a minute. And why DNA has a genetic code for the protein that needs to be made, but proteins are made by the ribosomes. So the ribosomes have to read it. And the ribosomes outside the nucleus in the cytoplasm, so those little things. And DNA is too large to leave the nucleus. So what happens is that DNA, it's all wound up, it's really tight inside there, but it can't come out these holes of the nucleus. So it needs to make these small strands of RNA so that it can actually leave and head out to the ribosomes who are out there in the cytoplasm, okay? But RNA can leave the nucleus because it is single-stranded. It's not this double-stranded helix, okay? Part of DNA, basically, just like the replication, it will unzip, just like when we talked about when it replicated. But now, instead of the whole strand kind of unzipping, just a portion of it unzips. Okay, and is used to template or assemble a complementary nucleotides into what we call messenger RNA. So they are the messengers that are going to go outside the nucleus and go to the ribosomes. Now, there's a little bit of a difference in these nucleotides when we go to RNA. So remember when we talked about DNA, it went from A to T, okay? A and T paired, and C and G paired. Well, here in mRNA and RNA, instead of a T, we actually have what we call a U, which is uracil. And so A now pairs with U, T pairs with A, and still G pairs with E. So these two pairs still sit there, but we also now have the A to U pair here in RNA, okay? And so mRNA then goes through the pores of the nucleus with the DNA code and attaches to the ribosome. So here's our DNA inside our nucleus here, okay? And we get mRNA mature just means it's been transcribed. So it becomes RNA, it leaves the nucleus here, and then it heads out to the ribosome, and it starts going through the ribosome, and the ribosome starts reading it, okay? And then we'll talk about the next kind of step, which is translation, okay? So the whole entire transcription is from DNA to mRNA, and then the mRNA leaves and heads to the ribosome before we get translated. So now we're at the ribosome. We have our RNA here, okay? And now we have to decode it. What the heck does it mean? Into a protein is called translation. So now we need to know what protein you need. So a different type of RNA, which we call tRNA, which is transfer RNA, carries those amino acids from the cytoplasm to the ribosome. So they will actually read the pairs in 3 parts. So they read them in groups of 3. Okay, and so what they do is they go, okay, here's A, U, G, and remember, it's now mRNA to tRNA, so we still do have that A to U. So A will go to U, A pairs with this U, and C pairs with G. Now, this code, this UAC code, when it connects in here, will code for a certain protein, just like this one does. So UUC, of course, will give us a tRNA of AAG. This is what we read, so UUC, and when we look at, I'll show you how we figure this out, it will code for a certain amino acid. Here, if we have AAA, it codes for lysine, and that's a certain amino acid. So those groups of 3 actually tell us what amino acid to bring, okay? And these amino acids come from the food we eat. Proteins we eat are broken into individual amino acids and then simply rearranged into new proteins according to the needs and direction of DNA. So all this stuff goes into your body, you digest it, you break it down into the certain amino acids, and then we turn it in, our body turns it in to whatever DNA has coded for, okay? And these are called, these 3 adjacent bases that I just talked about that tRNA actually comes to are called codons. So a series of 3 adjacent bases in an mRNA molecule codes for a specific amino acid called a codon. So whenever I say codon, I mean 3 bases off of messenger mRNA messenger RNA, and that will be our codon. And each transfer RNA or tRNA has 3 nucleotides that are what we call complementary, which means they match, to the codon in mRNA. And each tRNA codes for a different amino acid. And we'll go through and practice this a bunch of times, okay? And so this is what this means. So CGG codes for this specific amino acid off the tRNA. And this one, this is the codon on the mRNA. And the code, the 3-digit code or 3-letter code on the tRNA is what we call the anticodon. Okay? So this is a whole picture of kind of the whole kind of process. So mRNA carrying the DNA instructions and tRNA carrying amino acids meets in the ribosome. Okay? So remember, we're in DNA. Okay? We're going to go ahead and we're going to transcribe that messenger RNA, the one that's going to carry our messages, to our ribosome here, who's going to start reading it or translating it to the tRNA, who's going to come and bring those amino acids to code, okay? All right. So, and then the amino acids are joined to make a protein. So as you can see this, and you'll see in a minute, CUG comes and tells it to have this amino acid, this yellow one, and then the next tRNA comes in to match this 3 base pair on RNA, and they bring a certain amino acid, and it attaches to the first one. Well, this one gets released, and they start making what they call a polypeptide assembly. Remember, that is just the polymer that we call, it's called polypeptide, and the common name we use is protein. Okay. All right, so let's look at how this works. Okay, so you're going to use one of the codon charts on the next page, you'll see in a minute, to find the amino acid sequence coded for by the following mRNA strands. So the first one we have is CAC. Okay, and so we want to transcribe or translate that. All right, so what you're going to do here is you're going to transcribe each of these codons, which are in mRNA. So this is the mRNA, to an anticodon, which is tRNA. So this is just changing to tRNA. Then use the codon chart on the next page to find the amino acid sequence coded for by the second mRNA strand. So just on this one. So we look at these, we're going to transcribe, we're going to translate these into tRNA, but then we're going to look at this strand because it's going to tell us what tRNA is going to come get, okay? All right, so the first one, remember, it's a little bit different from DNA, and I'll give this one to you. If you remember, C pairs with G, but remember here in RNA, A doesn't pair with T because we don't have thymine anymore. Now we have uracil, U. And then C pairs with G. So I want you to pause for a minute, see if you can get the other 3, and then I'll go over those with you. All right, so hopefully you did that. So the second one should be GGU, okay? The third one should be ACC, and the last one should be, oops, I made that one wrong, so give me just 1 second to fix that one. All right, so I think I fixed that one, so let's look at that one again. That last one should be ACU. See, even I make mistakes. Okay, the last one, let's go ahead. Why don't you try all 4 of those? Try to do those really quickly on your paper and see if you can get them. All right, hopefully you tried. First one should be UAC, and then it would be UUG, CUG, and AUU. So that should be, these are the tRNA strands. But when we're talking about amino acids, we don't read from the tRNA, we read from the mRNA. So let's look at this second one. So we're going to look at this mRNA strand. I'm going to teach you how to use the chart. So this is a chart that we use to figure out what proteins. So this is how this works. So you always look at the first one, first letter in the middle. So our first letter says A. And remember, this is mRNA. So A. And then the second letter will be U. So then I'll go to the U here. And our third letter in that first one says G. So then I will go to the G here. So A, then U, then G. And that will code for methionine. So that'll tell me to bring that amino acid. Let's look at our next one. A, and then C. And that would be asparagine right here. Let's look at our third one, GAC. G, A, and then C would tell me aspartic. Let's see if I can get it to come. Aspartic acid. And then our last one here, UAA. So we'll start with U in the center, then go to A. That tells me a stop. So that means that's the end of that polypeptide. Okay. All right. Now remember, the big picture here is that we go from dRNA, we do transcription to mRNA, and then we translate to a protein. Okay. All right. So I want you to go back and process your notes. You're going to highlight any important notes, make connections to any of your notes in the left-hand column, write any questions you may have in the left-hand column, and then use your highlights, connections, and questions for your summary. And then at the end, write a 3 to 5 sentence summary. If you have any questions, please let me know.